Hello learners, welcome to my channel Botany V4. In today's video, we are going to study fungal septa. Now, when I say the word fungus, what comes to your mind? Yeast, mushroom, bread mold, right? The yeast granules that we use for baking and the highly nutritious mushrooms that we eat are all forms of the fungal thallus. If we study the thallus of fungi, we find that it can be either unicellular or multicellular. If we observe yeast under a compound microscope, we see that its thallus is unicellular. But in case of fungi such as Rhizopus, Agaricus, and Fusarium, if we cut a piece of the thallus, tease it slightly, and mount it, under the compound microscope, we observe an interwoven mass of thread-like structures. This mass is called the mycelium, and each individual thread is called a hypha, plural hyphae. If we magnify further, we can see that the hyphae of some fungi are hollow cylinders. Of course, they contain cytoplasm along with multiple nuclei. Such hyphae are called cenocytic, and the mycelium is called cenocytic mycelium. However, in other cases, generally in higher fungi, these cylindrical hyphae are divided into compartments. These compartments are formed due to the development of cross walls or partitions at irregular intervals. These partitions divide each hypha into compartments or cells and are called septa, singular septum. The word septum is derived from a Latin word meaning hedge or partition. Next, let's understand the formation of septa. The development of a septum starts with the deposition of a ring of new wall material along the inner edge. This ring gradually grows inward towards the center. This process is called centripetal development. Septa can be divided into two types based on their permeability: complete and incomplete septa. Complete septum is also known as non-perforated septum, and incomplete septum is also known as perforated septum. In complete septa, solid walls are laid down to block all the movement. Incomplete septa have pores. These pores are vital because they allow the movement of cytoplasmic organelles such as mitochondria and even nuclei between compartments. Now, based on their origin, septa are of two main types: primary and adventitious. Primary septa are formed in association with nuclear division and are laid down directly between daughter nuclei. Adventitious septa are formed independently of nuclear division. Let's now study the major types of septa found in different fungal groups. We are going to study 5 types. First, it is solid septum. Second, simple pore septum. Third, sieve-like septum. Fourth, dolly-pore septum. And fifth, bordered pit type septum. Now let's understand what is solid septum. A solid septum has no perforations. It is used by the fungus to delimit reproductive structures or to seal off old or damaged parts of the hypha. Draw two parallel hyphal walls and a solid straight line connecting them. Label it as solid septum. Next is second type, that is simple pore septum. Simple pore septum is found in members of ascomycetes. The septum consists of a single central pore. Draw the hyphal walls, but this time leave a single clear gap in the middle of the cross wall. This represents the simple pore septum. Third type is sieve-like septum. This septum consists of many uniformly distributed pores across the surface, for example, in geotrichum. Now, instead of one large pore, Draw a cross wall with many tiny, evenly distributed pores. It looks like a sieve or strainer. Fourth is dolipore septum. First, draw the hyphal wall. As the septum approaches the center, show the wall becoming swollen and thickened, forming a barrel-shaped structure around the pore. Now draw curved bracket-like structures on both sides of this barrel. These are called parenthesomes or septal pore caps. In dolipore septum, the septal wall is swollen near the pore to form a barrel shape. The open ends are bounded by septal pore caps derived from the endoplasmic reticulum. These appears like parentheses and hence named parenthesomes. Dolipore septa are found in members of Basidiomycotina, excluding rust and smut fungi. Fifth type is bordered pit type septum. In this type, the pore is surrounded by an overarching bifurcation of the septal margin, resembling the bordered pit found in the tracheids of Pinus. This type of septum is found in mucorales and trichomycetes. So let's quickly summarize. Fungal septa are not just simple partitions. They exist in multiple forms such as complete and incomplete septa, primary and adventitious septa, and specialized structures like simple pores, sieve-like, dolipore septa, and bordered pit types. Each type is adapted for a specific function, whether it is controlling the movement of materials, protecting the organisms during injury, or maintaining internal organization. What's fascinating is that these microscopic structures determine how efficiently a fungus can grow, survive, and interact with its environment. So, next time you study fungi, don't just think of them as simple organisms. Remember, they have a highly organized and dynamic internal system. And here's a quick challenge for you. Can you identify which type of septum would be most effective during injury and why? Drop your answer in the comments. I would love to see your thoughts. If this helped you understand the concept better, don't forget to like, share, and subscribe for more such botany deep dives. See you in the next video.